Good evening, everyone. Welcome. This is the Alliance to Cure Cavernous Malformations February webinar. And I'm going to give everybody, or give folks a chance to start rolling in. And so while that's happening, I just want to remind you that there's some things that are coming up. One of them is support group. Support group is going to be happening this evening, immediately after the webinar. And that's a wonderful opportunity to chat about what you heard, discuss things. Darla Clayton is the person who runs it. And then there is another support group that runs on Mondays at noon and on East Coast time, 9 a.m. Pacific time. And then housekeeping for this. When you have a question that you would like to ask, you will see at the bottom of your screen Q&A. Please put the question there and not in the chat. I won't see it if it's in the chat and it will not get answered. So please take the time, click that Q&A button, write in your question, and that's how we will keep track of what questions are getting answered and what aren't. All right, I think we're almost to capacity here. Our guest tonight is Dr. Issam Awad, whom we've known for many, many, many years. And he is the University of Chicago's John Harper Seeley Professor of Surgery. He leads the neurovascular surgery program and co-leads the stroke and neurovascular care team. He's the director of our very first CCM Center of Excellence at the University of Chicago, and he is the chair emeritus of our scientific advisory board. And tonight he's going to be speaking to us about our new update of the clinical care consensus guidelines. He'll talk about that. We'll answer some of your questions. And then he will talk about the results of the Atorvastatin clinical trial. And then we will have more time for questions, we hope. So Dr. Awad, welcome.
Connie, thank you very much. This is really a pleasure because those are two projects that are labors of love that have taken many, many years to get to this point. So it's a pleasure to talk about them. Let me first talk about the guidelines. Let me go ahead and share my screen. And what I wanted to first do is I hope that you can see the screen. Yes?
Yes, we can see it.
Very good. So the first thing is to get used a little bit to what the concept of the guidelines is there are two aspects to the guidelines. There is the class of the recommendation and the level of the evidence. Why do we do that? That's because we all have opinions. Sometimes they may feel louder or stronger, or we believe in them more, or whatever. But even different people would agree what is a strong, moderate, or weak, and what is the level of evidence behind it. If it's just an expert opinion, or if there are the highest level of evidence, which are clinical trials, etc. So each recommendation, we put a class. And what the class means is that 1 is for something and 3 is against it. So 1 is a green light, meaning go do it, you should do it, you aim to do it. 3 is harm, you should not do it, you should think hard about doing it. And then the 2 is in the middle. And then there are levels within those that we'll talk about, 2A, 2B, which have to do with whether whether we believe it is reasonable or it might be reasonable to recommend something. And then on the level of evidence, this is a lot easier, because then we look at what is the evidence that comes from more than one clinical trial. Then it deserves level A. We have a few things that are level A. Most of the things we have are level B or C because they come from clinical series. Some of them are better than others. And some of them are limited data, so we call it LD. And some of them are just BS, expert opinion. I believe this, you believe that, but that's okay. We still take those in consideration because experts have opinions for a reason. And then these guidelines are developed based on the published literature. So if it's not out in the literature, we can't consider it. And then it is also the result of what we call a consensus process. So we have a group of people that I'm going to be showing you in just a second that actually wrote these. Sorry. So basically, you're talking about many people who are surgeons, excellent geneticists, excellent uh, neurologists, who are either members of our clinical advisory board or who were recruited because of their unique expertise, like Dr. Paul McCormick, because of spinal vascular malformations and so forth. But it has, top surgeons, Dr. Lenzino, Dr. Smith, etc. And also, these people have to all agree on a recommendation. So it goes through a process. We call it the Delphi process, where if somebody doesn't like something, they say it. It is anonymous, so we don't know who didn't like it. We can tell sometimes, but we try not to guess. And we address the concern till there is a consensus, so that these opinions basically are both based on the literature, that are weighed and classed, and there is broad agreement among the experts on them. Why is that important? Because you need to separate those recommendations from a lot of crap that's out there and a lot of opinions that are out there that are not based on anything. And the two are not the same, the two are not the same. And you have a lot of stuff on social media and on the internet, and they are not weighed like this stuff is weighed. So that is very important. Let me, before further ado, just tell you a little bit how the guideline process was done. So the last time we did the guidelines was published in 2017, but it was based on literature through October 1, 2014. So between 2014 and 2023, there were an additional 2,672 publications on cavernous malformations that met some keyword criteria. And then we went one by one through those publications. We actually had 2 fellows assigned to this who went through it, and we oversaw it. And we went through each one of those papers, and we selected 234 papers based on their pre-articulated reasons why we should consider them for recommendations. So if there wasn't clinical trials, if there were guidelines by the Epilepsy Foundation, if there was a very good clinical cohort study, all of those were required to be kept. So the stuff that we didn't keep, if it's a study in mice only, or if it is one case that said I did this, or it's about a technique, I like to use laser, I don't like to use laser, we only kept those if they had outcomes differences that were addressed. So we were pretty strict, and then those 234 were not the only required ones, but they were required to be considered, and the writers had the ability to pick other papers among those 2,600 if they feel they are important. And then we came up with 53 management recommendations. There's obviously not time to go through all of that, but I'm going to walk you through it like you're flying over a field to kind of see what they look like. And then we're going to agree to take some questions from you. But again, there is not going to be time about each one's case and how they fit in these. So I've asked Connie to kind of keep the time and the priority of the questions so we can address them. So 18 recommendations of the 52 were class 1. They favor doing something. About half of the recommendations they favor with some reservations, and we had several class 3 which are do not do something. So that is also important. And then the levels of evidence, as I told you, level A, which is the most strict with 2 clinical trials supporting it, only 3 recommendations met that. They were mostly for things related to seizure medications, where there were big trials there for it. And then many were level B, but there were also a lot that were level C, meaning the evidence is poor, but we still kind of agree on it. So it's still better than anything random out on the internet, but the evidence could have some scientific errors, and we are all needing to learn more. So let me now go to the actual content very quickly and walk you through it. So basically, genetic testing recommendations. There is one class 1 evidence, which is that a history of family history is needed, and then who should be offered genetic testing. And we have also class 1 evidence about a variant. If you have a variant that is pathogenetic, meaning we know the mutation in your family, the person should be counseled about autosomal dominant inheritance, and we should identify at-risk individuals in the pedigree. So there is class 1 evidence that you are entitled to this information, and the centers that we accredit should all apply this standard of care. However, whether you do testing in family members or not depends on the choice of family members, but there are some recommendations that are given in this regard. Let me go to the next one, which is some recommendations regarding imaging. We continue to believe that an MRI for CCM should include susceptibility imaging as a minimum, should, if possible, be 3 Tesla. But we cannot mandate that because that's not widely available around the country for everybody. But obviously, if you want a better count of the lesions, it should be 3 Tesla. And then the class 1 evidence is that MRI is necessary for diagnosing and following cavernous malformation. So there is no disagreement on that. The class 3, which is do not do it, you do not get catheter angiography for a typical cavernous malformation. People have been hurt by this, they've gone blind, they've had other complications from it, and we feel very strongly about this. But if there is a legitimate differential diagnosis of AVM, then you get an angiogram. So I think this is helpful for the general doctors out there that may knee jerk and decide to go to an angiogram once they see a cavernoma. The other class 1 evidence that's important is about follow-up symptoms, follow-up imaging when there are symptoms. So this is very important, and also that in patients with prior radiation, when they have neurologic symptoms, an MRI must be done to look for radiation-induced cavernomas. So a lot of our class 1 evidence is in imaging. It's going to disappoint you because you want class 1 evidence on treatment, but at least we have that. And now, recommendations regarding surgery, they did not change very much, but they were expanded. I'm going to tell you a little bit about where we are. We still continue to not recommend, do not do it, a surgical resection for asymptomatic stable CCM if it is in an important part of the brain. We continue to allow some consideration for people to resect a single lesion if they have it, if it's asymptomatic, if it makes a particular difference in psychological burden or follow-ups and lifestyle or career considerations. But it's a weak evidence, level C evidence in favor of that. And asymptomatic for the specific goal of pregnancy is not recommended. So you don't get your cavernoma out just so you can get pregnant, because cavernomas are safe in pregnancy, and we will talk about that later. So these are helpful recommendations. We continue to have strong recommendations for surgery for cavernomas in the setting of seizures, and for symptomatic, easily accessible cavernous malformations. And we compare the surgical risk to how much life ahead you might benefit from it. Obviously, for a brainstem cavernoma, the risk is higher, and it's equivalent to about 5 or 10 years of living with the lesions. Now, radiosurgery, we continue to recommend it only for symptomatic lesions that are not that have unacceptably high surgical risks. There is uniform opinion that that is a reasonable option, but not for familial cases because of the concern about forming new lesions in familial cases, and also not for asymptomatic cavernomas. So that is a no. And laser ablation was now added as a new class 2B evidence that it can be considered for lesions causing seizures, with weaker evidence in larger lesions or those with hemorrhage. So the one where there is a strong evidence is for seizures. Regarding pediatric and older patients, we added some specific recommendations here about it being a little bit more aggressive in children and a little bit less aggressive with advanced age. So the level strength changes a little bit. Spinal cavernomas is a totally new set of recommendations. There are a lot of them because this is the first time that we did it. But what you basically have to realize is that we do not recommend surgery for asymptomatic spinal cavernomas, and they should only be considered for surgery if they extend outside the cord or are causing multiple disabling symptoms. There is a new relatively strong recommendation about post-surgical imaging follow-up, that early MRI should be considered to make sure that the lesion that was resected was actually taken out or not, and then ongoing surveillance is reasonable with annual MRIs for at least 5 years, particularly in pediatric cases. The medical recommendations are mostly things related to anti-seizure medications, being class 1, to not stopping seizure medications during pregnancy. That's a class 1. We also have some recommendations about the use of non-steroidal anti-inflammatory, which can be used with cautions as long as the lesions are non-hemorrhagic. And the issue about genetics we already discussed, but it was stated again here with regard to disclosing the diagnosis to a child, and this has to be made as a decision between the parents and the genetic counselor, with class 1 evidence for that. And the issue of genetic counseling prior to pregnancy is important for familial cases, so you understand the inheritance risk. And another class 1 evidence is folate supplementation for people taking seizure medications. Otherwise, recommendations about pregnancy with normal obstetric management, unless there are complications or problems. And there is also a good evidence that you should use aspirin or antithrombotic drugs if it is needed for heart or other condition, and the lesion has not bled recently. So this is also a new recommendation. And also with people with acute ischemic stroke, the benefit of giving the clot busting medications and blood thinning medications exceed the risk in cavernous malformations. We added some recommendation about being cautious about the use of oral estrogen and progesterone. This is a new class 3. Do not do it. So we have now more mounting evidence, although the level is still not a clinical trial, but there is consensus about avoiding the use of hormones. Statins remain elective. They do not do any harm, but they can be used if needed for other medical indications. And a statin should not necessarily be considered for decreasing bleeding in cavernous malformations. The same with propranolol. It is safe and tolerable, but the idea of using it to decrease bleeding in cavernous malformations really doesn't have any significant evidence. We have class 1 evidence that you should not have low vitamin D with cavernous malformations. And then if you do not know your level, then it's probably better to take vitamin D. So that's kind of where we are. We did have also a recommendation that aerobic activity is reasonable for patients with cavernous malformations. We do not address extreme activities beyond normal things. This is, I think, as much as I want to say. It is, as I said, just like flying over the field, but maybe we have time for 2 or 3 questions before we go into what many of you really wanted to hear, which is about the Atorvastatin trial. So, Connie, I turn it back to you.
Yes, thank you. We have, would you like to stop your share for the
Yes,
question?
sure.
We have just a couple questions right now that are related to the guidelines,
Okay.
as opposed to other topics. How might these recommendations be influenced by the possible changes to access to care, delay of treatment, delayed access to treatment, barriers to care, and insurance changes?
Yeah, this is very, very important, and we all are struggling with this. So as you all know, with Obamacare, it often opened up a lot of insurance to people who were uninsured, but at the same time, it put them in boxes as to where they can get their care. So many people sign up for these exchanges only to discover that they cannot go to the place they want to go, or to even a center of excellence or a clinical center. These most clinical centers and centers of excellence are committed to try to do something to work with you for out-of-network access, but it is not guaranteed, and this is a problem with our healthcare system. So the next best thing is, if you cannot and you're locked out, pending the next open enrollment, because once a year you have the option to get out of that box where you were put and pick a center that allows you to go to UVA or University of Chicago or wherever you want to go. Don't get locked out without knowing it and take some food stamps in exchange, because they give you free food stamps, but then you can only go to Saint elsewhere, and you want to be careful about that, because you already have a problem that can benefit from clinical centers that are accredited. So I think the other thing I would say is that other doctors ought to be able to read those guidelines and hopefully follow them, and we will hold them to it even if they're not in our guidelines. So you can tell them about the guidelines.
Thank you. All right. Given this is a spinal question,
Yeah.
given CTMs cause intraparenchymal bleeding, I knew I could say that, rather than SAH, what is the role of lumbar puncture for CSF assessment in this new guideline?
There is no role for it. We don't even bring it up because it's not even something on the table. No role for it. Maybe it's a good thing for us to bring up next time and to put a class C. No, do it.
Won't do it. Okay.
Exactly.
All right. Vitamin D supplementation is typically recommended with cavernous malformations. Should we be taking vitamin K along with the vitamin D?
There is no evidence that vitamin K will help or harm. There is possible concern about vitamin K increasing clotting, and that could be harmful. So I do not recommend vitamin K to be taken. Vitamin D, I recommend it in lieu of checking levels, you know. So I tell the patients in my clinic, it's an insurance policy that you don't have to worry, is my vitamin D fluctuating or low? So you take it a cheap 3,000 units a day or 4,000 units a day, and then you ensure that your vitamin D will never be low enough to harm your cavernomas.
Is there any clinical evidence, or do the guidelines address medical cannabis?
No, we did not address it, and there is actually no papers on it. So if there were, in particularly related to cavernous malformations, there is anecdotal evidence. So it's similar to the type of data we have with, say, ADHD drugs, stimulant drugs. There is anecdotal evidence. There is also Botox. There is anecdotal evidence of harm, but we don't have actually strong evidence to all agree on. Cannabis, the main problem we have is there is now evidence that it decreases cognitive function over time. So if the cavernous malformation is going to affect your cognition anyway, we would very strongly urge you not to take another 10-point hits on your IQ because of cannabis use.
Yeah. Okay. Is there consensus on and I'm going to put some interpretation on the direct question because I think I know what they mean, on the potential for rehemorrhage of a brainstem cavernous malformation, and how does that factor, excuse me, into the discussion of observation versus surgery
Yeah,
versus?
this is very carefully brought up in the guidelines, and once the guidelines are published, they should be published in March. You will, I don't know if you have a date, Connie, but you know, we haven't seen the proofs, but they were accepted, and we're working through the final form that they should come out. Once they come out, they're going to be posted for free. You can download them on the Alliance to Cure site. And there is a consideration for that. So that what we're talking about is the risk of surgery is weighed as to the number of benefit years you're going to gain by the surgery. And we're going to have a whole session on that, but the guidelines do address that.
Okay. And there were several people who asked about this 5 to 10 years with the brainstem cavernous.
Yes.
Can you just quickly, because there were several.
Yeah, let me tell you what that means. That means if you take the operation up front, there is a chance you're going to be harmed by the surgery. That harm by the surgery will not be exchanged by the benefit of no bleed because you had the surgery for another 10 years. Because if you were harmed by the surgery, you're in a wheelchair or you have a tracheostomy, you're going to have 2 or 3 bleeds to give you the same harm. So those 2 to 3 bleeds would not occur for 5 to 10 years. So you're taking all your risk up front, you know, and that's how it's weighed. I hope that explains it as best as we can.
Yes, and I think that at this time we should move on to the atorvastatin,
Sure.
and then if there are leftover questions now and we have time, we will take them at the end.
Great. So let me share the screen again and close this and open that. Okay, you all can see it?
Yes.
Okay, so we're gonna go pretty quickly. These are the slides that were presented at the International Stroke Conference 10 days ago, and 2 weeks ago, actually. And the paper that will include this data in great detail, exhaustive detail, is gonna be coming out in March, so in the next couple of weeks. So this was sponsored by the National Institute of Health. We did have some donated money to help us with developing the trial smartphone app. Why did we use symptomatic hemorrhage as the indication? This graph basically tells the tale. If you have so little bleed rate over 5 years, you cannot beat that with a drug. So if you brought people with very little risk of bleeding, then there is no drug that can beat that. So we chose symptomatic hemorrhage cases because they have a rate of hemorrhage over the next several years, including the first couple of years, that we hope we could beat with the drug. The other reason, the other thing that is important for this trial is it was based on very, very strong animal data. A lot of work supported by the NIH over many years. We first tried ROCK inhibitors that are not available in humans currently, and they work very well. Then we checked weak statins like simvastatin, and that doesn't help. But atorvastatin at a dose equivalent to 80 milligrams per day in humans, it's 80 per kilo per day in the mice, it does the same as the fasudil drug, and it chills down the bleeding in the cavernous malformations and decreases the size of the lesions. So it's block the ROCK effect, that atorvastatin has an effect of ROCK inhibition in addition to its well-known cholesterol benefit. Now, this is important. I'm not going to spend a lot of time on it, but it takes hundreds of patients for us to have an effect of a drug on the bleed rate, even in a group that bleeds at a very high rate. So even if the rate of bleeding is 15% per year, which is probably the highest group we have currently anywhere. Look how many patients it takes for us to have a 50% reduction effect. And if you want 25% reduction, look how many patients you need to prove it. I mean, those types of trials, nobody is going to do unless you know the drug is very, very likely to work, because they are very costly and will take a lot of time and will probably lock up all the patients in the community. So hence we need a surrogate outcome. We're not going to bore you a lot with this, but the surrogate outcome that we developed for this and we tested thoroughly and we got it approved by the FDA and the NIH is the amount of iron in the lesions measured by MRI. QSM study, and it looks like when you have an increase in the all cases that have a bleed will have an increase in QSM. And of those that do not have a clinical bleed, there are many cases that have an increase in QSM. Therefore, we have a lot of opportunity to test an effect on iron deposit, requiring less patients than if you were only counting symptomatic hemorrhages. So the math on this works out that if you have about 40 patients per group, and you were to lose 15 patients because they don't follow up or their MRI doesn't work out, you're still left with 25 patients per group. This allows you in 90% power to detect only a 20% difference in QSM. And this is calculated going both ways, meaning an increase will be detected with 90% power and a decrease will be detected. This is very important because we went into this trial with power calculations. So the hypothesis was that if the QSM increases by a drug, that means that drug is making you bleed in the lesion. And if the QSM decreases, then it is a bleeding benefit. And the 20% effect that happens would be statistically significant and detected. So that's kind of how the study was done. The study was done in Chicago because at the beginning of the study, the QSM was only accredited in Chicago. Now we can use it at multiple sites, so future trials don't have to be done in only one place. But even though it was done in Chicago, people traveled from all over. I'm very proud of this map, and we paid people for each visit. It wasn't a lot of money. It was a couple of $100s for travel, but it helped people get engaged and make it, and we did that. we did great enrolling Hispanic and Latino and African Americans, and we published that the African American patients were as likely as anyone else to stay in the trial and to complete it. So this whole fallacy that, you know, minorities do not do well in trials, we did not see that. So I think it's very good. We're very proud of that. We had 80 patients randomized. We had to screen about 326 subjects to get those. 41 were randomized to get atorvastatin, 39 to get placebo, and some people either did not follow up, it was COVID, they got pregnant, or they bled and had surgery. So we were left with 31 in the placebo and 33 in the atorvastatin that contributed QSM data for our primary outcome. We still counted bleeds in everybody. So this is what we called the intention to treat cohort. So we counted bleeds, we measured Rankin score function on everybody, but a fraction of those were the ones that contributed QSM data. So are these 31 the same as these 39? We tested that, and you will see that. So basically, compliance was excellent. Look at this compliance. You know, almost everybody had more than 90% drug use during 2 years verified. Only 4 people did not, and those 4 people actually were not among the ones who stayed in the trial. So they were really not compliant and eventually did not follow in the trial. So everybody who's in the trial took the drug. The trial was very well balanced for a lot of things. We looked at vitamin D levels, we looked at every other aspect of the disease, and it was very well balanced. But equally important, there were no differences in cases with and without QSM contributions, meaning the 31 that contributed QSM were the same types of patients as those who didn't. We were very careful about balancing vitamin D use, sex hormone, making sure they were balanced, and even propranolol, we wanted to be sure that there was not a great deal of imbalance, so that later you don't say, well, the effect wasn't there because one group took vitamin D and the other didn't, which happened in the Italian trial. So the primary outcome, this is really where the money is. The primary outcome was no different. So basically what happened is that it very slightly favored atorvastatin. So there was very slightly decrease. This effect size is 10%. So it is half the 20% that would have taken to be significant. And it was not significant because of this big uncertainty. So basically, this is what it was. And then we did some sophisticated analysis of the missing QSM data. And even when we impute those, the effect size is about the same. The effect size is about the same in every one of the 2 years of the trial. So it's not like you got tired from the drug and it quit having effect. So the primary outcome was neutral. It didn't do any harm, but it didn't do any significant benefit. We measured permeability in the lesions, and this also did not work out as being helpful at all. Now, this is important. People want to know this, meaning how often did people have actual bleeds? So the bleed rates in the trial were actually quite low. They were 15% to 18% over 2 years. So this is less than 10% a year in a group that was supposed to have 15% bleed rates. So why did they not bleed as much? Maybe because they had care at a center of excellence that gives them vitamin D and make sure they don't take birth control pills. But I don't know. But at the very least, there were no more or no different. There were 6 bleeds over 2 years in the atorvastatin group, 7 in the placebo group. Now, if you had only stopped at 1 year, you could have concluded falsely that the atorvastatin is helping. Look at that. But a month later, it started to catch up, and over the second year, there were much more bleeds on the other group. So at the end, they were about equal. So we're very careful about this over exuberance when you only have 1 and 2, because the next bleed eliminates the difference. So be very careful about interpreting this. But at the very least, atorvastatin did not cause more bleeding. There is no difference in bleeding events. And then measures of harm. There was no serious adverse events requiring discontinuation of the drug. There were no death in the group. However, there were a couple of adverse events that are due to a fall in one patient that we don't know if it was due to atorvastatin or not. She remained in the trial and continued. And another one had an elevated CPK, and then they chose to stop the trial and not continue, although we recommended that they take a drug vacation and resume the drug. They did not want to do that. So those were two effects. There were more adverse events in the atorvastatin per patient, which were the common ones that you would imagine: some leg pain, some diarrhea, and some tiredness were more common in atorvastatin than the placebo subjects, but they also were very common in the placebo. So, you had a lot of people with placebo telling me, I am tired and I have diarrhea. These are things that we looked at. Cholesterol was lowered by the drug like it was supposed to, but this is critical. The effect on Rho kinase inhibition in the blood was not observed. So this is a big lesson here, which means that the atorvastatin dose that was given did not inhibit Rho kinase in the peripheral blood. We have no idea if it inhibited it in the brain or not. But why is this different from the high cholesterol patients? Because the high cholesterol patients start with very high Rho kinase, and then atorvastatin inhibits it. But young, healthy people started with very low Rho kinase activity, which atorvastatin did not inhibit. The effect on permeability in the brain, not in the lesion, was present. So the atorvastatin was working on the brain permeability, but it did not break through to cause a benefit in the lesions. So at the end, this is a subgroup analysis that I want to tell you a little bit about. We talked, this was the harm, sorry. This is a subgroup analysis. So there were 3 pre-specified subgroups that we indicated ahead of time that we're going to look into. Why is this important? Because you cannot go look at subgroups later on and saying, I want to look at the ones with high MRS and the ones with big lesions and whatever, unless you pre-specify it before. Because if you check enough things, you're going to get something that sticks on the wall, But we initially said we want to see if there is a difference in the effect in males and females, whether there is a difference in brainstem versus non-brainstem lesion, and in sporadic versus familial cases. And the only potential difference here was that in the brainstem lesion, the effect was much stronger. And if the whole trial was only done in brainstem lesions, it would have been beneficial. So this, if you doubled the number of patients and they were all brainstem, then this effect would be more than 20%, and the difference would be significant. So what does that really mean? It means potentially that non-brainstem lesions got operated and taken out of the trial. A lot of people who had bleeds with serious lesions did not enroll in the trial. They were much more likely to enroll in the trial if their bleeds were small in the supratentorial space. But in the brainstem, they were more likely to enroll despite their lesions being large and their hemorrhage being significant. So What that means is that in future trials, we have to look at both groups and do what we call a stratification and futility analyses. It doesn't mean that you only should do the next trials in brainstem lesions, but it may mean that brainstem lesions will give you an easier signal to detect the benefit of the drug. So, in conclusions, and I know I'm going very fast because I want to leave time for questions. There was no concern about the safety of atorvastatin in CCM patients after a recent symptomatic hemorrhage, and no increased bleeding by any measure. This is the first study that can give you a serious clinical trial reassurance that if you're having to take a statin for cholesterol indications or any other reason, your heart or your stent, it's not going to harm your cavernoma. So we know that for a fact, and that will have a higher recommendation in future guidelines. But there is no benefit in decreasing bleeding risk as we had postulated, and therefore we cannot justify using atorvastatin for decreasing bleeding in CCM. So if you're using it for something else, you stay on it, may or may not help you, but to use atorvastatin to help your cavernoma, we just don't have evidence that that is the case. The issue about the weak ROC biomarker effect basically make us ask a very cogent question. Would the stronger specific ROC inhibitors that are being developed by pharmaceutical companies achieve the desired benefit because atorvastatin was simply a weak ROC inhibitor? That is a possibility. The direction of point estimates in the subgroups suggests the need for what we call stratification and adaptive design in future trials. So at the end of the day, I want to just take a little moment, and I think this slide is as important as any. The group at Johns Hopkins with my co-chair, Daniel Hanley, collected all the data and ensured data quality. We used a very sophisticated data entry system with specific definitions for cavernous malformation that were already harmonized in prior trial readiness studies. The biostatistics were awesome. This was a difficult trial in the terms of the statistics. When working with Lancet Neurology to get it published, we had to answer about 39 pages of statistical questions, but it got through it because we had very, very good statistics. A trial manager, Agnieszka Stadnik, and trial nurse, Justine Su in the more recent years, and Christina Piedad before that. Kelly Fleming was our independent safety monitor. She was looking at the bleeds and all the harm and giving us signal whether the study should stop, and obviously did not do that. We had other safety consultants who were available for help. We had a statin expert who would answer the patient's question about whether their leg pain or elevated CK levels or whatever. The ROC assays were done by the inventor of the ROC assay, Dr. James Liao, and a huge bioengineering effort by Tim Carroll and Kincaid with the QSM data. We had many students that worked on this, but the fellow I want to recognize the most is Roberto Alcazar, who actually is a co-first author on the paper because he did just amazing work day and night during the summer and fall analyzing the data, and during the year prior to the completion of the trial to prepare it so that we could report it as quickly as we did. Connie and the Alliance to Cure, and we could not have gotten patients from throughout the country without the support. The CASH Consortium with all the centers of excellence that were part of the trial readiness helped us get ready for this, and StrokeNet as well. So a lot of people to thank. But this trial basically has opened the road because in terms of safety and oversight, it's important, but we did a lot of preclinical and imaging research that's going to be used in the cavernous angioma field by other people in the future. And it's going to, the idea of novel clinical trial design that allows you to detect an effect with less patients becomes extremely important for innovation and infrastructure. So we're very proud of what we did here, even though the effect on bleeding was not proven, but we showed a lot of other important data, and you will be able to read about it in a couple of weeks. So I'm going to stop share, and I think we did pretty good with going over a lot of stuff and leaving time for questions, Connie.
We did. You did. Yes, thank you. Um, okay, so one participant in the trial is wondering if they'll find out whether they were taking the drug or placebo.
Yes, so we have a letter that is approved by the IRB. We had to approve a letter before. We can't send you a letter, believe it or not, without being approved by the IRB. But the IRB requested that we not send it until the paper is published. So you will all receive a letter that's already approved by the IRB shortly after the Lancet Neurology paper appears. In that letter, I summarize the results in lay language, like I tried to do here, but in lay language. And I also give you the option to send us an email or a text, and we will let you know which group you were in. But we also explain to you in the letter what it means for continuing to use statin, not continuing to use statin. So all of that will be in the letter. And we're not anywhere away. You can always reach out to us. You know, the AdCash beeper will work, the AdCash email will work, and you never were shy reaching us if you were in the trial. So you can reach us anytime, and we'll be with you till you're happy that you understand how you contributed and what it means to you as a participant. The greatest thanks is to the participants.
Someone is wondering whether there were any adverse events or side effects that were specific to CCM that you wouldn't see in just the general
Not
population.
at all. Not at all. However, uh, there were a little bit more of the myalgia and fatigue in relation to placebo than in the large statin trials. So we did review the large statin trials, the 40,000 trials, and those have very slightly more. There's almost as much leg pain in the placebo as in the atorvastatin. But in this population, maybe the CCM patient population, or maybe because they're younger, are more sensitive to the effects of the atorvastatin. Of course, I can't prove that because we did give it to healthy young subjects, but you are young and you don't have high cholesterol, so maybe the drug gives you a little bit more of these, but it didn't affect your compliance. Not even one patient asked to be on the half dose, which was an option, you know? Yeah, one patient asked, but when I told him yes, he said, no, I don't want to be in the trial anymore. But that was it. But everyone else stayed on the full dose. So very good.
Do you think there may ever be an opportunity for a brainstem specific atorvastatin trial?
No, I honestly don't think so. I think, just the funding for that has to come from NIH, and to justify doing a whole trial over five to 7 years in brainstem. And I think personally, I would want to see a drug with stronger ROCK inhibition before I put the community through that.
All right, here's somebody with an obvious scientific background, which is why I've held this question a little bit. Given the limited efficacy of atorvastatin, do you think that despite the ROCK pathway being considered central to CCM pathology, another pathway, such as VEGF, could play a more dominant role in humans? If so, could this justify clinical trials with Avastin or COX inhibitors?
Yes, and yes to all of that. But the problem with Avastin and the other drugs are the greater side effect profile. So Avastin is not proven safe in CCM. And it does cause people to bleed in the brain, from our experience in glioblastomas and others. And therefore, the willingness to go to that will have to be, you'll have to accept the greater side effects. So, and my experience with this group of patients who I've gotten to know very well, it's like getting them into your living room, is most patients, even if they bled from their cavernoma, they don't want a lot of side effects. They don't want a lot of side effects. I'd have hours of discussion as to whether the slight lightheadedness should make them stop the drug, and we talked about that, and they would stick it out. But just what I see is that you take much stronger drugs. Now, you could conversely select the very aggressive lesions, maybe with biomarkers and whatever. We select the group that has had 3 bleeds in the past 6 months, and they have no other options. But how many of those do we have? And can we do a trial in that? So, yes, other targets could be useful. We need to select the patients most likely to deserve the side effect of that drug so we can try it.
I think that's the end of the questions that I see for atorvastatin. There have been a number of people, and I'm going to go to a more general topic now, who have been asking about, with the change in administration and some of the changes that have been announced, what do you see as the impact on CCM research?
Yeah,
And
that's very sobering, right? Um, there are the government will honor the contracts that they've already offered, but they will back out of the indirect costs that they had committed to, because they have the ability to do that. That's been challenged in the courts. For people who don't know what the indirect costs are, is the money that goes to the university that gives me a lab and the place to work. So my lab, I don't charge the NIH the space in Chicago downtown for my lab, per square foot, or nor do I charge the internet, nor the library use, nor the computer storage facilities that we have. So those are called indirect costs. So it's being challenged whether it's fair for the government to cut those out, because those are real costs. So the way I could see it is that eventually they get turned from indirect costs into line item costs, so that the next grants we would have to say is, I'll have to say, well, I need $45 a month for internet, and I need $280 per square foot for space, you know, and then you'll give me more direct funds. Whether the government, how much money is the government going to save when they do that, I don't know, but they may put us through that. They're in a mood to do that, so they may do that. We have to be prepared. They may even cut the whole funding structure of the NIH, so that trials like this one that generate not only the clinical question, but knowledge to do the next trials. These trials, this study and the studies that led to it, the biomarker studies, the mouse studies, basically were about $30 million of research that is in the public space for any drug company to take all this data and do what they want with their drug. No drug company would have spent money to develop QSM for this indication or this. So I think, we've done a lot of public service with it. But I think where it's gonna come now, and people who have skin in the game, and it's gonna be people who have a drug that they want to make money on the drug, and I hope we've given them enough tools to be able to go for it with a promise of benefit.
um, hold on a second, because I got caught up in your answer and I was forgetting to look at the questions. Well, okay, so the second part of this is the indirect costs. There's a whole other thing going on with staffing in the NIH and FDA, CDC, all across HHS. Is there already visibly an impact?
Yes, they don't answer the phone anymore. I mean, I don't know where they are. So it's, I think they are also going through the stun factor. But on the other hand, I just submitted a grant due February 5 to the NIH. It was accepted to be reviewed. So whether we'll have a review or not, we'll wait and see whether they'll have money to look at it or not. But we're going to continue to play the game, and it's not a game. It's a very important thing for our advances. None of the knowledge we have that will drive the next therapies could have come out without the basic science that took into to get us to this point.
Last question. What motivates you, Dr. Awad, to dedicate your life to this? Small question.
Well, thank you for asking this question. What motivates me is the look on the face of every patient I help. And we help patients in many different ways. We help patients by telling them this past year was okay, or I had a baby and it's okay, or you had to go into my thalamus and I'm okay. So all of that. And even the people who are not okay, knowing that they have a partner and an advocate to help them. So that's what keeps me going. But what really keeps me going is, and I will say this, I've told it to you, as long as I'm able to do it and I'm good enough to be allowed to do it, I'm not going to quit till we have a significant progress that help our patients.
Thank you. Thank you. And we appreciate that incredibly.
Thank you.
Yeah. So
it was a great hour. I think we covered a lot of things.
We did. Let me remind people that if you check in the chat, you will see a link to the support group that meets right after this. You might have to go up a little bit because now we're getting all the thank yous in there to Dr. Awad for this wonderful presentation. And we will have our next webinar will be on March 6th, and we will be hosting Chris Gibson, the CEO of Recursion, who will share the information about the outcome of their trial. So it's back to back. It's really wonderful and exciting for us.
It is. Thank you. Bye-bye.
Thank you. Thank you, everyone. Have a wonderful night.